as today's speakers are Revell, demonstrating his package phytools, and Klaus Schliep, demonstrating his package phangorn. Liam is originally from Canada. He is an assistant professor in the Department of Biology at UMass Boston. He got his PhD from Harvard in 2009 and advised by Jonathan Losos, followed by a postdoc at Nessen. Klaus is currently looking for a job. His PhD advisors were Mike Kendi, Barbara Holland, David Pinney, and Peter Waddell, and he just finished a postdoc in Paris with Philippe Lopez and Eric Baptiste. So I'm very happy to have them speaking today.
Okay, so thanks to Eric for organizing this and for inviting Klaus and I to talk today about phylogenetics in the R statistical computing environment. So today I'm going to talk about the capabilities. I'm going to focus on the capabilities of the phytools package. Phytools is our phylogenetics library. I'm the author and maintainer. Really, most of phylogenetic tools in the R environment are sort of built on core package, a really important core package written and developed and maintained by Emmanuel Parody, who sort of literally and metaphorically wrote the book on phylogenetics in the R environment. And this package is APE. And in fact, when Eric invited me to talk, I suggested that Emmanuel would be I don't know him personally, but I thought he'd be a more appropriate speaker. But unfortunately, or fortunately for myself, because I'm always happy to promote phytools, he wasn't available. So today, I'm going to so really in this sort of hour, it would be possible to learn how to use all of the R phylogenetics functionality. But there's lots of online resources available to learn more about how to use R and how to do phylogenetic analyses in R. Just to give a couple of examples, of course, there's the R project homepage, and then this is the website for the contributed packages repository CRAN. And there's also a CRAN phylogenetics task view that's maintained by Ryan O'Meara. Let's see if this will pop up. Whoops. Yeah, so and this is sort of documents all, well, it's not comprehensive, but it covers a lot of the functionality in different R packages, R phylogenetics packages, as well as sort of like a nearly comprehensive list of the phylogenetics packages in R. There's also a really helpful RSig Philo email listserv, and this is just the website for the listserv. But all the previous inquiries are archived, and so you can see by thread various discussions on different topics. And then if you're interested in phytools in particular, I document a lot of my development activity as well as sort of errors and bug fixes and things like that on my blog, which I'm always happy to direct people towards. Oops. And here, for instance, I'm documenting the fact that I'm giving a phyloseminar today. But in general, I might sort of respond to user error reports, or document new functions, as well as sort of my sort of development of those functions in some cases. All right, I somehow got to the wrong. This is actually my last prepared slide, because this sort of seminar is going to serve more as a software demonstration than a traditional seminar. The major functions of phytools fall sort of broadly into 5 categories. I'm going to talk mainly about 3 of these today, and I'm not going to cover comprehensively what phytools can do in each of these categories, but the 5 categories are simulation, phylogenetic comparative methods, and visualization, plotting trees, etc. And there's also some functions for tree input, output, and manipulation of special types of phylogenetic trees, etc. And there's a couple of functions for inference, but Klaus is going to package phangorn is a much more important package for phylogenetic inference, and so I'm not going to talk about the very small, the few functions that are in phytools for inference today. And I believe Klaus is going to talk about the phangorn package, which is inference oriented. He just appeared. So just to sort of as a manner of introduction to the R environment, it's a command line driven environment. So this is actually the way the Windows GUI for R looks. You can also run R from the command prompt, or you can run R in Unix and Windows. It looks like, or Unix and Linux and Mac OS, and it looks sort of slightly different in those cases, but the prompt remains the same. It's a command line driven environment. Oftentimes, well, I don't say oftentimes, but sometimes users interested in some of the functionality of my package, but not familiar with the R environment, will send me an email, for instance, saying, what file format does, your X function in phytools require? And actually, what I'd say in general is that this used to be obviously a common problem when I write computer programs to do phylogenetic analyses in C, for instance. In that case, we had to be very specific about what file format was required by the function. But in R, the paradigm is different. In general, the analyses, the sort of input-output of data and trees are handled by different functions than the analyses, and the analyses are performed on these objects that are created by those functions and then stored in memory. And so just to give you a quick example of that, here are a couple of different tree file formats. So this is, for instance, this is familiar to most people. It's a sort of it's a Nexus tree file, and then this is sort of the same tree, but stored in it sort of wraps across many lines here, but stored in a Nuix style, or just sort of standard Nuix style, like you might read in for instance, in the program Philo. And we can sort of read in either of these trees equally in R. And so I'm just gonna to show you this, I'm just gonna load the ape package. One second. Little and so here I'm reading in just the sort of standard phylop style tree, and then I can plot it. I can actually do this, using a function in ape called plot philo. And here the labels kind of aren't displaying very well here because of the scale, but and I'll show you another function that could fix that in a second, but generally we have the tree. And the same could be done using an alternative function called read.nexus. In that case, we'd be reading in the nexus file. So if I wanted to apply that, I would just do this, and you can see that I have the exact same tree in memory from these two different input formats. In phytools, a lot of the functions pertain to comparative biology, so the analysis of phenotypic trait data, and so we need to get trait data in as well, and that can be read in from various formats. So for instance, a common format would be a plain text file, common to limited. Whoops, I just sort of opened that in Excel by accident, but like this, and we can read these data into R easily as well using a base function. And so there's our data. Okay. For the purposes of illustration of the functionality of phytools, I'm actually going to not use this data set. I'm going to use simulated data mostly, and also because phytools has all this sort of simulation capability. So to get going with illustrating phyltools, I'm going to load the phyltools package, and I'm also going to load Luke Harmon's package called Geiger, and that's just for the purposes of simulating some types of things in this talk. And so we've loaded both of these packages now, and now I'm going to simulate a pure birth tree on the and I can visualize that. Actually, I'm going to visualize it using phyltools tree plotter, and so you can see that tree here. All right. Now, just for the purposes of subsequent simulation, I can rescale the tree. I'm actually going to use a function called rescale tree that exists in the Geiger package again. I'm going to rescale it to have a total length of 30 in this case. And now I'm going to use a phyltools function for simulation that's called fastBM. And fastBM does, well, it was originally designed to do fast Brownian motion simulation, but it actually now does a whole bunch of different types of variants of Brownian motion and other types of continuous character simulation on the tree. So with it, we can, we provide the function fastBM with the phylogenetic tree, as well as instantaneous variants of the Brownian motion process or evolutionary rate. And actually, fastBM can return the simulated states at internal nodes on the tree. And so I'm going to do that. And so you can see that these data now are stored in the vector X. And the vector X contains, in elements 1 through 50 in this case, the tip states. And those are, the tree is a simulated tree, so it just has numbers as tip names. So in elements 1 through 50, it contains the phenotypic states at the tips of the tree. And then 51 through the end are states and internal nodes from simulation. And so I'm going to show another, this is a new function of phyltools, actually not in a released version of phyltools, called phenogram. This is actually very similar to the Picante, rather, is another phylogenetics and macroecology our package, and it has a function called traitgram. This is a function called phenogram, just for distinction, that just has the option of including the states and internal nodes. So if we know what the states and internal nodes are, say because it's a simulation or because we have other sorts of information, we can include those. And so in this case, I'm going to plot that. So this is a phenogram. I've just dropped the tip labels by turning them off. And so this is just a phenogram of Brownian motion on the tree, and the phenogram is, the trait axis and the time from the root. And so that's the realized Brownian motion simulation, including the states and internal nodes. I just visualized it using phenogram. If in a more realistic sort of situation, I would have only the states 1 through 50, the states of the tip nodes of the tree. In that case, it'll actually use maximum likelihood to estimate the ancestral nodes in the tree. And it actually uses a function that also belongs to the phyltools package to do that. So that's why it takes a little bit longer. And here we see that we have the tip states plotted, and they're connected to ancestral nodes that are estimated. All right, so that was pretty easy. So I'm going to try out some other capabilities of fastBM. So we can do, for instance, we can do simulations with bounds and with a trend. So first, I'm going to show what happens when we do a simulation with a bound. I'm going to impose like pretty tight boundaries. I'm also going to return the internal states. And so in this case, you can see the effects of the boundary is that there's a lot of reflection off the bounds. It also means that our ancestral character estimation will tend to be really bad because we're estimating under a strict sort of pure Brownian motion model, and that's not what we've simulated. So just to illustrate that, I'm going to do the phenogram, plot the phenogram, but in which I only provide the function with the tip states, so it has to estimate the states at internal nodes. And so that'll take a second. There it is. So obviously this is a sort of radically different view of the history of the phenotype in the group, because the model that we've used to reconstruct ancestral states is wrong. But it provides a pretty neat visualization. We can also simulate with a trend, and you can imagine sort of what's going to happen then. The trend parameter is mu, so this is just the mean of the Brownian motion process per unit time. I think that's it. And I'm going to plot a phenogram of this too, including the ancestral states. And you can see the effect of the trend is really dramatic. And finally, if we compare that to what we would get for ancestral states if we ignore the trend, the difference is really quite dramatic. Okay, I think that pretty much covers what I was going to do with FastBM. So I'm just going to, the tree originally had a length of 30. I'm going to rescale it to 1 just for purposes of convenience. And then I'm going to show, what I'm going to show is discrete character simulations, because phytools can do these too. And actually, it's a little bit more than that, because rather than just simulate the states at the tips of the tree, it also simulates the transitions between states along the branches. And so here what I'm just constructing is a matrix that's a instantaneous transition matrix for the Markov mutational process for the character, or the discrete character that we're simulating here. And I can use a phytools function called simHistory to generate a character history under this model. And then I can visualize that history. I'm just going to do some preliminaries here. I can visualize that history using another phytools function, which I'll show you in a second. Oops. And that's called plot simmap. And here I've chosen the 3 colors, blue, green, and red. And what's illustrated is the history on the tree. Plot simmap is kind of has a lot of different options too. We can visualize it with different, with or without these dots indicating the transition points along edges. And you can see that in this character history, you know, for instance, the character might transition from green to red to blue to red. I apologize for anyone that's red-green colorblind for my choice in colors. All right. Now, just for point of illustration, if we wanted to also count the number of changes that occur on the tree, we can do that pretty easily just by knowledge of the structure of the simmap, the mapped tree in history, the simulated map tree in history. And I'm not really going to get into how this works, but we have 27 changes, and so if we were to go about and count every single change in this tree, 1, 2, 3, 4, 5, etc., it would correspond to that number 27, because that's what we've counted. So if we wanted to set a constrained simulation so that we had a specific number of changes in the tree, we could do that too. We would just write a loop around this and reject any simulated histories that didn't have the correct number of changes, or a number of changes on a range, for instance. All right. So with phytools, we can also simulate the history of continuous character that in which the rate of evolution for the continuous character depends on the discrete state. So now we see that we have a tree in which we've stored the condition for a discrete trait, say green, red, blue, etc., on the tree. And so what we can also do is we can use that tree and we can simulate on it, but in which the rate of evolution depends on that state. And so just to illustrate this, I'm going going to simulate with 3 different rates, 1, 5, and 10 for the 3 different states, which are 1, 2, and 3, and I've forgotten which colors those correspond with. I'd have to go up and look at that again. So this is sort of just a matter of preliminary here. And then the function that I'm going to use is called simRates. It's another phytools function. And it'll simulate the character evolving under this process in which it has a rate that's dependent on the state for the discrete character. All right, and then I can plot this. So I'm going to plot 2 different things. I'm going to plot the simmap tree, and then I'm also going to plot the phenogram as well. Actually, we can do even better than that, because we can retain the internal states with simRates as well in the current version of simRates. And so this is probably going to replace my upper plot, but you get the idea. I'll just reverse them here. So it's kind of neat, and maybe in the future I'll add the possibility of coloring the phenogram by the condition for the discrete state, because then we could really have the potential for visualizing the I've actually changed the color scheme here. I apologize. I just defaulted back to the default color scheme because I didn't enter it. But all right. Okay, so there's a number of other simulation functions in phytools, but I don't really have time to go over them now. Phytools also includes a number of different likelihood and Bayesian phylogenetic comparative methods. And some of them are just too computationally intensive to go through, but there's a number that I can illustrate, so I'm going to show a couple. So first, I'm going to take the data that I just simulated. Actually, I'm just going to take the first 50 characters, or basically the tip states from that data, because remember I simulated the internal states as well. So now I just have the tip values. And the tree that I simulated before. And actually, let me close this. And what I'm going to do is I'm going to fit a model in which I'm going to fit the evolutionary rate. I know what the rates are. I know what the generating rates are. What I'm going to do is I'm going to fit a model which the rate of evolution is different on the different colored branches of the tree. I simulated under that model. Now I'm going to fit the model using likelihood. And this model is, this method was developed by Brian O'Meara in 2006 evolution paper, and I've implemented it in the phytools package in a function called Brownie Light. So it's maximum likelihood. Actually, you can see that I'm running this for the first time, but it had a couple of false convergences, which sometimes happens. But it got over those, and you can see that here are the estimated rates. So for instance, the generating rates were 1, 5, and 10, and the estimated rates are 0.62, roughly 5, 4.7, and 14.8. And so that's pretty good, because we only have, it's pretty close to generating values, because we only have a fairly small number of tips in our tree. We only have a 50 taxon tree. And the function also ran pretty fast, but it actually is quite computationally intensive, because it involves inversion multiple times of a n by n matrix. But that can be avoided, actually. And I recently wrote a Revell version of this, so a restricted maximum likelihood version, which runs much faster. And the difference is not going to be super apparent, but you can see it basically ran instantly. And here are the estimated rates using the restricted maximum likelihood. They're not exactly the same, but they're quite similar. And they're actually, in theory, they should be unbiased, whereas these will be slightly biased. Normally, though, in this case, I'm fitting this model knowing the true generating history. But normally, we actually just for the process, I know that when and exactly when the discrete character changed over time. But normally, we wouldn't know that. Normally, we'd have to estimate it from the data, and we'd estimate it from the tip states for our discrete character, and we've actually stored those in our tree. So here's a vector containing the tip states for our discrete character in the tree. And one way to deal with this is by generating possible histories, sampling possible histories according to the probability. And this is called stochastic character mapping. And there's a phytools function for this, true. Yeah. So I'm just going to generate 100 stochastic character histories using make sim map. And this just takes a couple seconds. There's a couple of neat functions. One is I can compute the overlap between these histories and the true history. And, oops, I misspelled it now. And this just gives me this will give me a vector that is the overlap between the mapped histories, the stochastic mapped histories, and the true mapped history. And you can see it's they're modest in some cases because the character changes quite frequently. And we can also run Brownie-Revell on all of these. I'm just going to show this really quick. And I'm actually going to use sort of a trick to just return the estimated rates rather than the whole output from Brownie-Revell. Whoops. I misnamed the thing earlier, so this will take a second. So I'm sort of being encouraged to wrap up. Here's what we get by averaging the rows, and so you can see that the states, their estimated rates again are not as close to the true rates as when we knew the true history, and that's not sort of unexpected. But they have the same general trend. So there's lots of other functions in the phytools package. Phylogenetic signal estimation with and without sampling error. There's some new functions in which we can fit evolutionary models using Bayesian MCMC, incorporating sampling error, well, basically incorporating variability among the individuals for species intraspecific variability, and then there's a number of other plotting functions in phytools. And you know, for development of a lot of these things, I encourage people to check out my blog, as I mentioned before, phytools.blogspot.com, and I try to be pretty diligent about updating the blog with all the updates to the phytools package. And of course, I'm always happy to get you know, more users for phytools, and so people can contact me if they have any questions about the use of the package, and I'll try to respond to you as quickly as I can.
Now it's my phangorn package, and phangorn, of course, the name comes from or is inspired by the phangorn forest, and of course phangorn forest is, at least for Lord of the Rings, in New Zealand, and as several people have been probably to the Doom meeting, so it's quite a close idea to come up with a package which reconstructs phylogenetic trees, so it's several trees, so it's kind of a forest. So phangorn, I thought, is a good name for it. And the package started kind of as part of my PhD, and everybody who knows David Penny knows that he's normally a list with 100 things to do in phylogenetics, and he gives each of his PhD students several of these topics, and they're normally quite diverse. So that's how phangorn started off. And I started building it really some maximum likelihood function and some parsimony functions on top of the 8 package, and so I wasn't quite sure how much people know about R, so just want to kind of tell that there's quite a lot of R packages devoted to phylogenetics, maybe something around 20 different packages, and on the R website there's a kind of a task view, phylogenetics, which tells a little bit what most packages do, and there's also a mailing list where people can ask questions about specific packages, or also about questions they have to do, and maybe get some hints which package they can use to do. So that's probably a good thing to get started. And then I'm just put in one first slide to how to kind of get a little bit help about R packages if you don't know much about them. So of course first you have to install them, then you start a package, and then there exist several forms of how you can get help, and you can type these commands in and you will get some different type of help, which is quite nice. Is so-called vignettes. That's kind of a document which tells you a little bit how some functions work, and this is kind of, this talk I prepared is also kind of the form of a vignette, and it's kind of a nice thing that you can write these commands and be actually computed while you compile the documents. So they are really, what you see is what you get, so kind of thing to do. And so the first thing, of course, is you read some data into the first, and normally you have data in a Nexus format or in a fillet format, and so that's what I'm reading here. Is now just some data, and most of them haven't really written in much functions how to read in data, but use mostly apes function and then convert them to a different data format, because for phylogenetic analysis I sometimes need some different format than the format Emmanuel uses in ape. And I also have some other convenience functions to get data out of other objects, especially kind of matrices or data frames, which are common objects, or also to convert data from a type called alignment, which is from another R package called seqnr, or these DNA bin objects, which are the standard data format in ape. So, and if we've done kind of reading in the data, which kind of here I did with these function read further, I can also kind of combine sequences. So I just read in 2 genes, and then I'll write cbind, and then I'm combining these sequences. And the thing is, these sequences I read in here were all kind of very closely related species, so they actually were samples of the same species. So sometimes the sequences are exactly the same, and if the sequences are exactly the same, we kind of get problems when we want to estimate trees later on. So we can use a command unique, and then we have only one, then we delete all the duplicate sequences. Yeah, so of course, similar, of course, we can do it with many other packages. Also, I can have a function called simseq. If we don't want to read in sequences, we can simulate them ourselves. And here I have just a small example for this. So I'll first generate a random tree with 5 taxa, and then I just say, yes, simseq, I want some sequences taking this tree. I want to have lengths of 10, so I want to construct 10 characters for each tip. I have here a type equals user because I want to generate user-defined type where I have levels of 1, 2, and 3. I could also write just type equals DNA or type equals AA for amino acids and would just generate amino acids or DNA sequences. And there's now even a package called Phylosim, which does a lot more complicated simulation for these character-based, especially DNA sequences. So there's a lot of different packages. Each of these simulators has maybe there's advantages or disadvantages. So this one is just there because it's kind of nice if you have it in your own data format, and so then it's easier to use. So then, of course, if we have some data, then we can we want, of course, produce trees, and if we have the simplest method is of course distance-based methods, and to have to construct a tree from a distance-based methods, we have first to compute some pairwise distances, and there's several functions which can do this. So here I've kind of given 3 different versions of this. The first one is just DNA, so this is an aped function, so I convert my data first into this aped format and then just use the functions around, and you can use some different models like Jukes Cantor, or then the next function is a function from phangorn. It's just using the Hamming distance, or if you with these character-based things, it's just looking if you have 2 sequences, how many differences do they have, and then also what's one maximum likelihood version where you can specify a transition matrix and some base frequencies if you like. Otherwise, it's just something like a Jukes Cantor model. So, and then we have a distance matrix, and then we can just use some ape or phangorn functions and construct some trees. We can construct a UPGMA tree, we can construct a neighbor joining tree. There's also minimum evolution models from Gaskell in ape. So there's kind of, we can use one of these distance matrix and construct 3 different trees. And then, of course, the big question is, of course, are these trees the same or are they different? And so we can compute the Robinson-Falls distance between 2 trees. Robinson-Falls distance computes the number of, or is the number of differences between of the internal edges between 2 trees. So and we see kind of that the neighbor joining tree and the minimum evolution tree are very similar, whereas the UPGMA tree is not so similar, which is not surprising. So yeah, and then of course we saw already a little bit, especially ape has really nice plot functions, so we can plot our trees. So I just plot the neighbor joining tree and then UPGMA tree, and also with these commands it's very, very simple, just plot. And I'm also just wanted to show you that these plot function can be very, very sophisticated, and it has lots of arguments, and you can just kind of change colors and edge widths and use different fonts. So this one is just some very, very basic plot without doing any special things, just using the default values. So now this was kind of the first thing, that was distance-based methods. Then the other kind of another popular method is of course parsimony, and we can kind of first start just evaluating the parsimony score for the trees we constructed beforehand, and for example we can type parsimony for these 3 and the data we have had and get the number back 239. So we have 239, we need at least 239 changes to would be necessary to represent these trees with these data. And of course there's some functions to find some better parsimony trees, so we can do some nearest neighbor interchange and try to find better trees, and there's a function called parsimony ratchet or p-ratchet which kind of tries to find or to minimize the number of the parsimony score. And if we run this function, then we kind of find a tree which has kind of a parsimony score which is 1 lower than the one we got from the neighbor joining tree. So it's very similar, but of course we get some improvement. Yeah, and of course then we can use this parsimony score and also kind of can construct, for example, for the parsimony, some ancestral states, and we can kind of take a root. Of course, parsimony trees are normally unrooted, but we can kind of define some root and see what's kind of would have been the root sequence, for example, of kind of the first 20 taxa of this data, and then we can kind of plot this. Unfortunately, they are all very, very the same, so it's not as nice as I wanted to show the data, but we can see the kind of could show the proportions of different nucleotides. So that's done with the seq logo, which is kind of a nice package which kind of can show how the proportion of different nucleotides is. And of course we can also show something which is similar to beforehand, so we can show, yeah, how do we estimate the ancestral states, or when we use these parsimony criterion, and there's different ways to estimate these. This representation is not necessarily unique, what's red or what's blue, but that's kind of one possibility, and so one has some idea which ancestral states could be read for. Yeah, here we have a legend for A's would be red, and C's would be green, and G's would be light blue, and T is this violet. So we can kind of have an idea where the changes would have been. Yeah, so after maximum parsimony, kind of course, nowadays more often maximum likelihood is used, and so there's also one function to compute maximum likelihood, the maximum likelihood of a tree and for the data, and kind of we can start with a very, very simple model. We have had a tree beforehand, and we show what's the likelihood for this data, and we can get some, we just put in this, then we get some kind of result, and likelihood here is not necessarily very good. We have the unconstrained likelihood, that would be the kind of the best achievable likelihood when kind of when the expected side patterns would be really the side pattern we see. And here our model was so far, because we didn't define anything, it was just a Jukes-Cantor model, the rate matrices, all these non-diagonal elements are kind of one, so actually we only have to look for these rate matrices, see rate matrix to the upper or the lower diagonals, so just the off-diagonal elements. And also the base frequencies, when we don't define anything or we don't estimate anything, it's just equal base frequencies. So this, of course, so then of course if we want to use maximum likelihood. The question is, what kind of model should we use? And I've implemented a function which does model test, and the function I call just model test. So it's very, very simple. And we give this function the data set, and we can specify the models it should check, and then it kind of computes for these different models the likelihood, and also defines then the IIC or BIC, and I can use Jukes-Cantor or Jukes-Cantor plus invariant sites, and Jukes-Cantor plus gamma, and gamma plus invariant sites, and estimate here these 60 models. I have more models implemented, but it's just fitting as much on this page, so I just restrict myself to these 4. And the thing is, this primary function is actually quite generic, so we actually can define our own transition models. And so here is now a little bit of R magic, I would say, because when I compute these different models, the model I get back from such a function like this stores, this is just kind of gives you a result which is printed, but it actually stores normally in the back some information. It would store the tree and the data also, and all the model parameter. And of course, if you do this for each model, then you save a lot. Then you store far too often all the data sets or something. So I'm saving all these model parameters in some kind of hidden environment, and I can recompute then these model, and which would be a Jukes-Cantor plus I model. But now we want to estimate the substitution matrix differently, and so we actually want, for example, we define a substitution matrix where we estimate parameters. We give us one rate for transversions, and we give us 2 different rates for transitions. And why would we like to do this? Well, it could be, in our case, the species are very, very closely related, so we actually have lots of transitions but almost no transversions. And if we optimize them these likelihood using our kind of own transition model, we would get a AIC from 6,504, and if we compare this with these models, it's actually not a bad AIC. Kind of the one which are better are these GTR models here, which contain a lot more parameters. So that's actually quite a nice thing. And here we can see the parameter we estimate. So we have different base frequencies here, and here we see these kind of 3 different rates. Kind of the rate for the transversions is kind of fixed to 1, and then we see that there's kind of a transition transversion ratio would be something between these 2 values, because we have kind of 2 parameters for these 2 different types of transitions. So that's something which is kind of can be also quite easily done in R, and it probably takes some more effort to do in a package like Pulp or some other methods and some other packages, and sometimes it's probably not so open to play around with these things. So then, of course, these objects we get when we use this function PLM. I've written a few so-called generic functions, and so we kind of can use ANOVA PML, which kind of just takes different objects and compares them in a ANOVA-like table. We can just extract the log likelihood. We can plot these trees of them, which is of course using just kind of the ape function, but tells it what type of or element of these object we want to plot. Then, yeah, print PML is just a function which prints some basic parameters of these objects. Update PML is quite nice. If we have an object and we say, oh yeah, I'll estimate all the transition matrices, and now I have kind of different gene, for example, and I want to can, for example, just update my data, or I have a different additional species, so I can update, oh yeah, I just update my tree and the data and let all the other parameters the same, because if we optimize them, they will change only slightly, so we don't need to start optimizing all the parameters from scratch, so we can just start a few of them to optimize. And it also has a function which kind of computes the covariance of each of the edge lengths, which we can kind of do nicely if we have maximum likelihood estimates. So it's an approximation, and so we can do something like these image plots where we kind of show the correlation between all these. This is kind of, we have these trees here, so we have a lot of, I think it's around 40 species. So we have here around 80 different edge lengths, which kind of, on the diagonal we see the correlation is always 1, and there's a few elements which have also kind of correlation of 1 or -1, so they are very highly correlated. So it's kind of, if you have edges which are close by or next to each other, then if you make the one edge longer, then the other one has to be shorter, so that's why you get these negative correlations. Whereas when you have a big tree, let's go back to one tree, if you look at this edge and this edge, they are far away, so the correlation between these two edges will be likely 0. So there's a few functions which do a little bit, kind of, where you can play a little bit around with your estimates and compare them. Of course, if you have estimated a kind of a tree in your model, you want to do some bootstrap analysis and check how good your results have been, and if they're reproducible. And so there's a function who can do a little bit these bootstraps. So it has also here it's also you can use, if you have several cores on your computer, you can make use of them, so it can kind of spawn a little bit the computations, and it hopefully takes a little bit less time. And then quite often in R, if you're not used to it, you have these kind of ... argument, and this ... argument is always kind of you can define what you want actually to do with it. So it's normally you give commands to a function which is called internally by this function. So, and this function is just using these function optim PML, which kind of optimizes these PML objects, and then you can define what kind of things do you want to optimize. And down here I optimize my fit. That was kind of this PML object I had. I want to have 100 bootstraps, and I want to optimize, want to do some nearest neighbor interchange. So I'm just optimizing the topology, but I'm not optimizing for each of the bootstrap samples the base frequencies, for example, because I hope when my sequences are long enough, the base frequencies won't change much. And for bootstrap analysis, I'm mostly interested in the topology, so I'm just switching between the topology and hope that these other parameters are the same. But I could do some optimization of rates or base frequencies or invariant sites also with this function, but then of course it takes long. And then we can plot these tree again and plot also on the edges these bootstrap values. So we see there's the long edges normally have 100 on them, and if the edges are getting shorter, then the values are sometimes getting smaller. So that's kind of what we would expect. And there's not only the possibility to show these bootstrap trees, but I implemented also some consensus networks. That, of course, was one of the inventions of one of my supervisors, Barbara Holland. And so I wrote a function, consensus nets, and have a plot function for this. And consensus nets are quite nice, so you kind of see highlight a little bit the differences which kind of occur in your data. And if we go on the next page, we can see a little bit. There's here some kind of, it's not a tree anymore, but it's kind of a network, and sometimes we have these kind of small squares in it, and it's kind of done with a nice function, so we can actually have it as an RGL object and as a 3D object, and can turn around a little bit. And this looks already quite fancy, but of course now we have all the names, and if we replace the names with just some nice balls, it looks almost like some chemicals, and we have kind of can show these objects and rotate them and zoom in a little bit if we would like to, and see where are these kind of differences, or where do we have kind of between these different trees. So that's kind of a nice thing. So was kind of my main talk about phangorn. If there's questions, we can still go a little bit on the command line and show some more other things. So that was kind of more or less the main functions there. There's a little bit more hidden functions. And I also just wanted to mention that R is kind of quite flexible how you use it. You can process data in batch mode, so you can, yes, if you have 100 data set and want to make some analysis, you just write some batch things and send them away. You can use these interactive mode Liam has shown before, and of course you can also, R has quite some nice support for literate programming, and that was kind of how this document was done. And so this document is really written with the commands for the functions, and the plots were then all produced by the program when you compile your document. So that's really how you would do reproducible research, I think, or I think it would be nice to do it like this. And also you can produce nice slides and show what you're doing, and you have immediately the results, how it looks like. So I think it's quite nice. Yeah, and then of course, if you're on the search for some Christmas presents, just advertise Emmanuel's new book, because it's just new out, and so it covers a lot what's in different packages. It's, of course, Emmanuel Paradis wrote the A package, which kind of is the underlying packages under most of the other phylogenetic packages, but in his new second edition of his book, he also covers a little bit of phangorn and many other packages. The thing is, I think phytools is probably too new that it's covered in much of it in it. So that's, yeah, and of course it's always a problem with writing a book about R packages, is that R packages normally change much faster than the book. But so if you still need a Christmas present for your lab or yourself, it's maybe a nice read. Yeah, and of course I have to thank kind of my collaborators in Paris and from New Zealand, and of course all the people who sent bug reports or suggestions which improved phangorn. So thanks a lot.